briefly talk about malaria in the lab. So I'll go straight to the topic. And it's interesting, although we know malaria has been around for a long time now, and we know it had a major impact during the world wars, World War II, during the Korean War, and so on, as my colleague mentioned. It wasn't until the mid-late 70s that we were able to culture the malaria parasite in the lab in vitro. And if you look at the literature, you see a very dramatic change in the type of publication before, let's say 76, 78, and post. So all the data that was published before was based either on clinical samples and clinical observation, or on work that was carried out on macaca fascicularis or macaca rhesus monkeys. So it was only at the time when they were able to, for the first time, to culture the malaria parasite, Plasmodium falciparum, in the lab, that the focus of the research changed, and it changed very dramatically. And you can see since the paper by Trager and Jensen, many things have happened since, and now, as my colleague mentioned about the life cycle of the parasite, now because we can grow this parasite, and as I specified falciparum in the lab, we've been able to develop assays to screen for drugs for almost every stage within the blood stage of the parasite. However, this has been limited to Plasmodium falciparum. It has been very difficult to develop any culture for Plasmodium vivax. Now, we've talked mostly about falciparum today. Falciparum is the #1 Plasmodium species that's responsible for most deaths globally, but Plasmodium vivax is associated with the most morbidity globally. So vivax is prevalent in Asia, in India, Pakistan, all the way from Iran, all the way to Indonesia and the Philippines. And although it's not responsible for as many deaths, it actually has an enormous economic impact and impact on the well-being of society health. So interestingly, so far, nobody has been able to develop a continuous culture of Plasmodium vivax. There has been many, many efforts. The Gates Foundation has funded multiple projects to try to culture vivax in vitro, and it has repeatedly failed so far. So what I'm showing here is on the left is a summary of some of the many attempts at culture vivax, which have failed. And then on the right, showing pretty much what we can do, which is to grow vivax in the lab, not grow it, maintain it alive, maybe up to, in some cases, 5 or 7 days in the best of cases. So one of the approach that is being used globally instead is to use Plasmodium Synomology as a surrogate for Plasmodium vivax. So Plasmodium Synomology shares many characteristics with Plasmodium vivax. The two most closely related at the genetic level and also at the morphological level. So here I just summarize some of the similarities, well, genetics, life cycle, morphology. Probably the most important is the formation of what we call hypnozoids, which is a dormant form of the parasite in the liver, which is responsible for the relapse of malaria. This is what in the past was referred to as traveler's malaria, is when I suppose Westerners went to malaria endemic area, they will go back home, and maybe a few weeks, a few months, or a few years later, they will develop malaria, and that's why they call it traveler's malaria. And this is due to relapse of malaria. Now, relapse of malaria is associated, is only found in vivax and not in falciparum, but it's also found in Plasmodium Synomology. So Plasmodium Synomology presents a very attractive parasite to use as a surrogate for Plasmodium vivax for research. So We know that Plasmodium Synomology can infect humans. As Benoit just mentioned, this paper that came not too long ago, but there's another paper in JID recently where they find cases in Cambodia and in Malaysia. So we are finding more and more documents where we find Plasmodium Synomology in humans. Now, they're not associated with any acute cases of malaria, but we do see that they do infect humans as well. Historically, we have also used Plasmodium Synomology as a model to study pretty much every drug that's today in the clinic has gone through tests using monkeys and Plasmodium Synomology as a model. So Plasmodium Synomology has many aspects that are attractive to be used as a surrogate for vivax, and the morphology can be very, very similar. And in fact, in many cases, it's very difficult to distinguish the two in the laboratory. These are some original pictures from, I think, the 60s, year 62, where they show how similar the morphology of these two parasites can be. So as Kevin mentioned earlier, Synomology was the strain that was used to identify the pre-rhesuscitic stage of malaria. So that's before the blood stage of the malaria parasite. And the experiments were first done on Plasmodium Synomology and then repeated with Plasmodium vivax. And very early, at this point, it was suspected that this pre-rhesuscitic stage was associated with the relapse. And it wasn't until the 80s where it was demonstrated that you have the formation of what we call this gymnozoid, these dormant forms that are found in the liver. So following the success of the Plasmodium falciparum culture in 76, culture in the lab, continuous culture, in the early 80s, there were two success stories about culturing Plasmodium Synomology in the lab. The first one by Nguyen and the second one by Zhu showed two strains of Plasmodium Synomology that were adapted and grown in the lab for a couple of months in one of the cases. However, interestingly, these efforts were never able to be replicated until the last few years. So in the last few years, the Plasmodium Synomology was cultured in the lab again, and now it has become a standard, and many labs have the technique. And interestingly, it seems that not all Plasmodium Synomology are the same. And probably one of the reasons why the parasite was, people were unable to reproduce the results from the 1980s is because they were using different parasites. It's only until a few years ago that Alvin Shua took the same strain that was used in the original article from 1982 and managed to put the strain into culture by following the infection of a monkey. What you see here, this was the first experiment where this strain was cultured in the lab. What you see is that after you extract the blood from the monkey infected with Plasmodium Synomology and you put this into culture, the parasite is multiplying, and this is why you have this so-called split, because the number of parasite, the percentage of parasite is very high, and you have to split it so they can continue to infect red blood cells. And the second indication showing that the parasite is actually multiplying in culture versus surviving in culture is that you have the different stages of the parasite. So you have trophozoites, you have skeletons, and you have rings, even at the day 7. That means that the parasite is actually multiplying. So since then, this strain has been adapted to do type of molecular studies, lab studies, morphological studies, and so on. And as I mentioned, it comes from the original isolate that was isolated when it comes from Malaysia. The same strain that was first cultured in the 1980s. Somebody infected an Iotus monkey. An Iotus monkey is a monkey from the New World. It's an South American monkey. And then it was a tube that was amplifying this monkey that was used to infect rhesus monkeys, monkeys, from which the culture was able to be derived. Oh, okay, sorry. Thank you. So what this is showing is the different stages. So if you look at the right, you see the different stages of development of the parasite. Again, it mimics very much what we see today with Plasmodium vivax. And on the left, you see every time that the parasite multiplies in vitro, and again, the cultures have to be split because the parasite is multiplying at a very high rate. And in the bottom left, you see that the levels of drug resistance, so in this case is chloroquine, I'm sorry it's not written, in Plasmodium Synomology and vivax is pretty much the same. The IC50 here is equivalent, as you can appreciate, showing once again that Plasmodium Synomology is a good model to study Plasmodium vivax, which we cannot culture in the lab. Since then, these strains have been isolated as single clone, and this allows to do work, molecular work on this strain. And finally, just to finish off, so this model, the idea is to adapt this model to the drug screening that are specific for Plasmodium vivax. And what this is showing you here is on the bottom, you have Plasmodium Synomology, Plasmodium falciparum, and you have some reference drugs, the ones that we find in the clinic, showing the susceptibility to these drugs in the two different species of malaria parasite, falciparum and Synomology. And on the right is, again, showing more where you have some similarities or not. So one of the things that has been found is that, in general, most of the drugs, malarial drugs that target Plasmodium falciparum work on Plasmodium vivax. However, we now know that in many cases, the mechanism of resistance and maybe even the mechanism of action may be different. One of the best examples is chloroquine. We know that chloroquine, the mechanism of resistance in vivax, does not seem to correlate with the mechanism of resistance in falciparum, and it's possible that the mechanism of action may also differ somehow. So having a model, so the important part here is having a model, having something that you can culture in the lab allows you to address these questions. So it is critical to be able to adapt these parasites as for any other infectious agent, to grow them in the lab so that you can actually study them. And I think I'll stop there. So yes, if we forget to target the hemophobe, so the dormant form of malaria, this will continue to be a reservoir for malaria in the long run. So it could be possible that we manage to control Plasmodium falciparum in the coming decades or so. But as long as there is a reservoir, meaning a dormant form of malaria that we may be carrying in our liver, we will always have the problem of reemergence of malaria. And if we want to achieve the goal of malaria eradication by the 2040s, then dormant malaria, or Plasmodium vivax more specifically, cannot be ignored. So thank you. That's all.